All right. Hello, everyone. So we're going to finish off this unit by discussing chapter 20, which is enzymes. All right. So first, a brief introduction to enzymes here. Enzymes are proteins, but they are a specific type of protein. They play an incredibly important role in the cell, and that is to catalyze chemical reactions. Right? So an enzyme is just a protein, but specifically a protein that acts as a catalyst in a reaction. Catalysts are things that make reactions go faster. Okay. And so that's exactly what a enzyme does is it increases the rate of a reaction in the cell. It is not changed or consumed at all in that reaction. So a catalyst is something that's not consumed. And enzymes, likewise, after they get done catalyzing the reaction, they're just their old normal self again. So they do this by lowering what's called the activation energy of a reaction. So here we have this free energy diagram here, and this would be the energy of this reaction without a catalyst. The height of this barrier here is what's called the activation energy, and it corresponds to the rate of the reaction. So, so for example, let's just take a reaction which is converted, which is the breaking apart of a glucose molecule. Okay, that would happen spontaneously on its own without any sort of a catalyst, but it would occur extremely slowly, right? And if you're a cell, you want this to happen when you want it to happen, right? If the cell's trying to metabolize sugar, it's not going to wait around for a reaction to take place spontaneously. It wants it to happen right then and there, which is what an enzyme does. So it's by speed up the reaction, we mean that it can actually happen in a timely manner within the cell. Okay, so this would be the height of that activation energy without the enzyme. And then when you add the enzyme, it decreases the height of this barrier, speeding that reaction up. Okay, notice it doesn't change the energy of the reactants all by themselves. That would be this point here. This point here is the energy of reactants. Nor does it change the energy of the products, which would be the endpoint over here. This is the energy of products. So it doesn't change the energy of the reactants. It doesn't change the energy of the products. What it does is decrease the height of that activation energy barrier, allowing the reaction to happen fast. Okay. And again, by fast, I really mean in like a timely manner for the cell. Okay. Yeah. So again, they increase the rate of a chemical reaction by reducing the energy required for that reaction to take place for the conversion from reactants to products. Remember, if we're talking about enzymes being a catalyst of a chemical reaction, that means that there's a chemical reaction underlying each of these enzymes, some conversion, making and breaking chemical bonds, right? Conversions from reactants to products. So this is an example. This is a carbonic anhydrase. This catalyzes the formation of bicarbonate, converting carbon dioxide and water into this, which would be bicarbonate, and a proton. Okay, so this is a sort of a way of grabbing CO2 from the gas phase and putting it into solution, be the function of this particular enzyme here. And it does that by, creating and making the new chemical bonds. So this would be CO, this would be H2O, and then over here we have, right, so we've made new chemical bonds, we've broken and made new chemical bonds, converting from reactants to products. Again, this is something that would take place naturally, but if your cell wants to fix carbon, put it into the solution, right? Your blood and your other cellular fluids. It can do it on the fly with the carbonic anhydrase. Okay, so all enzymes catalyze chemical reactions in the cell. That's what an enzyme does. Generally speaking, most enzymes are globular proteins that have unique three-dimensional structures. So the difference between every different enzyme is going to, first be that primary sequence, that polypeptide chain, but then importantly, those fold differently to create unique three-dimensional structures. And these three-dimensional structures recognize the reactants of the reaction, which we call in biochemistry terms, we call the reactants substrates. Okay, the proteins these enzymes have what's called an active site where these substrates bind. This is just a small portion of a protein, right? So if I'm just going to do like a little sketch of some globular protein, it might have just one teeny tiny section that's the active site of the protein that's dedicated to binding that substrate. And within that active site, there are going to be specific amino acids, which residues are there to interact with the substrate. So we have these different amino acids, each with their own functional group, and so they can interact with those substrates through the same ways that amino acids interact with one another. We talked about the hydrogen bonding, the formation of salt bridges, or even those hydrophobic interactions might hold a substrate in place. Okay. Oh, so here's an even better example of what I just tried to draw, which is then a structure of an enzyme. This is lactase. Lactase will convert lactose into its two monosaccharide components, galactose and glucose. And so you can see that in this giant lactase enzyme, there is just one tiny section that is the active site that actually binds that lactose. So here's the lactose, the substrate pictured in gray, and you can sort of zoomed in, you can see the different amino acid residues that are interacting with that galactose. And so what this enzyme would do was catalyze the breaking apart of that disaccharide into its monosaccharide components. So the first step is going to be to bind that disaccharide glucose, and then this enzyme is going to catalyze the chemical reaction that's breaking that bond. How it does that is incredibly complicated. Okay, so we're not really going to be talking, that's way advanced biochemistry to get into the details of active site chemistry. We're not really going to be talking about those details here. We're going to learn sort of more topical stuff about enzymes. Okay, so like this is a good example of what the type of information that we're going to see in this chapter, and that is categorizing an enzyme as either the specificity of that enzyme as being either absolute, group, or linkage. So what do I mean by that? So specificity is how specific that enzyme is to a substrate. So lactase is an example of an absolutely specific enzyme because it only catalyzes that breakage of that link, that disaccharide linkage in lactose. It wouldn't work for sucrose, it wouldn't work for maltose, it wouldn't work for any of the other disaccharides that we learned. The only disaccharide that will fit into that lactase is lactose. Okay, so that's an example of an absolutely specific enzyme. Catalyzes one type of reaction for exactly one substrate. Let me actually use my color coding here. Okay, a group specificity is an enzyme that catalyzes one type of reaction for similar substrates. So for example, if there was an enzyme out there that did break apart multiple disaccharides into their monosaccharide components, but like regardless of what the identity of that disaccharide was, that would be have a group specificity. So the example here, right, so catalyzes for similar substrates. So the example here is hexokinase, which will add a phosphate group to any hexose, right? So glucose, galactose, any of those 6 carbon sugars, hexokinase can recognize and add a phosphate group to. So that would be a group, an enzyme with group specificity. And then the last one is linkage specificity, which catalyzes a type of reaction for a specific type of bond, okay? So the digestive enzymes in your stomach, for example, chymotrypsin catalyzes the hydrolysis of peptide bonds. Doesn't matter which peptide bond is it. Is it a alanine and a glycine or a tryptophan and a glycine? Doesn't matter. All it does is recognize that peptide bond and catalyzes the the breakage of that peptide bond, the hydrolysis of that peptide bond. Okay, so how enzyme catalyzed reactions work. First, the substrate has to bind to the enzyme. This forms what's called the enzyme substrate complex, right? So you have your enzyme, you have your substrate. First step is binding, forming that enzyme substrate complex. Okay, then within that complex, something kind of crazy happens at that active site that converts the substrate to a product. And again, that's the details of that are really complicated and going to be beyond the scope of this class, but we just know that they catalyze substrate being formed into product. And then you have your enzyme product complex that then dissociates and allows those products to leave, leaving you with your leftover enzyme just as it was before the reaction. Remember, reactions don't change enzymes. Okay, so this is an example using lactase. So we can say the first step is that we have our enzyme by itself and the substrate by itself. And in step one, we have the binding of that substrate, which forms our enzyme substrate complex. Then something magical is happening at this active site that converts this disaccharide bond into, cleaves it, hydrolyzes it. So now we have our sugars that were once linked together. Now they're broken apart. And this would be the enzyme product complex. And then the last step is for that product to dissociate from the enzyme. So now I have my two individual products, galactose and glucose, and I have reformed my enzyme all over again. Oops. Okay. Talking about the active site, there are two models for enzyme action. One's called the lock and key model. So this is the idea that you have a rigid substrate binding to a rigid active site, much like how a key fits into a lock. And then they have this induced fit model. So this is a more dynamic model where we have a flexible active site that adapts to the shape of the substrate. Okay, so the substrate's coming in and then the enzyme kind of forms around it like, like a fist grabbing something or something to bind that enzyme, that substrate more tightly. There are different examples out there of enzymes that are lock and key types or induced fit. I will say most enzymes tend to fall into this induced fit category. The ones that are more lock and key tend to be the ones that are not as specific. So those linkage type enzymes that we talked about, that ones that will just recognize any peptide bond, that might be a much more rigid active site that the peptide just kind of fits into perfectly, and that's how it's able to recognize it non-specifically. Okay, but so you can find examples of both of these models of enzyme action out there. Okay, so little learning check here. So which is true about the enzyme's active site? Is it the entire enzyme? Is it just a section of an enzyme? Or is it the substrate? So remember that active site is just the tiny region where the substrate binds. So the correct answer would be B, it is a section of the enzyme. Okay, and then in the induced fit model, what happens to the shape of the enzyme when the substrate binds? Does it stay the same or does it adapt to the shape of the substrate? In the induced fit, that is the model where it adapts to fit the shape of the substrate.